Okay, good morning, everybody, and welcome back to day 2 of our Invasive Species Forum. I hope you're all well rested and ready to jump into a full day of invasive species learning. Before we begin, it's important to note that the Invasive Species Center honors the long history of the First Nations, Inuit, and Métis Nation peoples on the lands now known as Canada and strive to show respect to their ancestors, their communities, and to them individually. We greatly appreciate the significance of the lands, waters, and all living things, and offer our gratitude to the Indigenous peoples for their care for and teachings about our Earth. Our head office is located on the traditional lands of the Anishinaabe, the Batchewana, and Garden River First Nations, as well as the longtime settlement of the Métis people in the Robinson-Huron Treaty area. We're meeting virtually again today too, so I'd like to acknowledge that the Indigenous peoples are the traditional stewards of the lands and waters where each of us attends the forum this morning. Yesterday, we had an announcement from the Minister of the Ontario Ministry of Natural Resources and Forestry about our grant program. So we're thrilled to say that the proposals for the next cycle of the Invasive Species Action Fund will open on March 6th. So if you're interested in applying for funding, please visit our website to learn more. We have a really exciting lineup planned today. Some of the highlights include our second special session of the forum, which starts at 10:50 a.m. In this session, you'll have the chance to hear all about environmental DNA, which is the latest technology in detecting invasive species. Our speakers will be shedding light onto the use of eDNA with a special emphasis on water soldier in Ontario, invasive Burmese pythons in Central Florida, airborne eDNA, and the newest eDNA collection technologies. Today, we're also thrilled to have Henry Lickers, retired International Joint Commission Canadian Commissioner, who will be giving a keynote presentation titled May We Live an Interesting Time, Indigenous View. You'll also want to stick around till the very end of the day because we have an interactive workshop on woodland-style florals and the connection to biodiversity, land, and reclamation led by Lucilla Ford. I already have all of my paper and colored pencils here ready to go for that art workshop, and I hope you join us ready to go with your materials too. So to officially kick off day 2 of our forum, I'd like to introduce a very special guest, Trevor Jones, who is the Parliamentary Assistant to the Minister of Agriculture, Food, and Rural Affairs. Mr. Jones was kind enough to send in some recorded remarks for this morning, so I'll just get it loaded here.
Hello, everybody. I'm Trevor Jones, MPP for Chatham-Kent-Leamington, and proud to be the Parliamentary Assistant to our Minister of Agriculture, Food, and Rural Affairs. Thank you for allowing me to join you today. Ontario is blessed with abundant natural resources, fertile farmland, beautiful forest cover, and pristine lakes, rivers, and streams. To preserve this treasure, we all have a part to do to be stewards of the land. Locally, in my communities of Chatham-Kent, Leamington, and Pelee Island, I know firsthand the challenges posed by invasive species, including zebra mussels, purple loosestrife, and of course, Phragmites. Through knowledge sharing, research initiatives, and collaborative projects aimed at managing invasive species, you're doing just that, and we thank you sincerely for your efforts. I can assure you, we stand united with you to manage invasive species and protect Ontario's natural spaces. One recent example is the emerging threat posed by the spotted lanternfly. In earnest, our ministry team, along with partners at CFIA, academic researchers, industry partners, all work together to collaborate and track its movement and prepare growers for the eventuality that this destructive pest is fast approaching our borders. To date, our efforts have included direct funding for monitoring, research, education, and mitigation efforts. We're continuing to consult with specialists at home and worldwide to better understand this pest behavior and prepare and discuss best practices for early detection and response. We know we have a very capable partner in you. Our government appreciates the role you play in protecting and preserving the well-being of all our natural spaces and the agriculture industry. I'm confident in the work you do, and I know you have a very and busy, productive day planned. I sincerely appreciate you taking a few moments to spend with me, and I thank you again for your efforts. Your participation today is a service to all of Ontario, and I thank you. Have a wonderful conference.
Okay, thanks so much, everybody. Thank you so much, Trevor Jones, and to Amafra for all the ongoing support. We are running a little bit early this morning, so we're actually just going to take a 10-minute-ish break. We'll start right at 9 just to make sure all of our speakers are ready to go. So maybe just refill your coffee, your tea, and sit back and relax, and we'll join again in 10 minutes. I'll leave this open. We can just sit and wait. Thank you so much, everybody. Okay, everybody, we'll get started now. Thanks so much for joining us. We have some great speakers this morning to join us in the first session of the day. This Aquatics Invasive session is sponsored by Ozero Solutions, which is a Canadian company whose mission is to protect the environmental, economic, and social wealth of our lakes. They offer innovative boat decontamination stations to prevent the spread of invasive species and makes the decontamination of internal piping possible. So I just want to say a big thank you to Ozero Solutions for the support. They actually won the Young Professional Award at last year's Invasive Species Center Awards ceremony, which is happening again on Thursday if you want to tune into that. So I'll pass it over to our first moderator of the day, Jen, who is our zooplankton diagnostics and research intern. She assists with a lot of different invasive species related program work, but primarily helps to coordinate the Asian Carp Canada program. Thanks, Jen. It's all yours.
Hi, good morning. Thanks, Maddie, for that nice intro. So like she said, my name is Jen Wagner, and I'm the zooplankton diagnostics and research intern, and I will be your moderator this morning for the aquatic invasive species session. So before we get started, I just wanted to remind everyone that there will be a brief question and answer period. So if you did have a question for our speakers, just put that in the Q&A box, and I will read them aloud to our speakers following their presentation. So starting us off this morning, I would like to introduce our first speaker, Andrea Weise from Fisheries and Oceans Canada. And with that, I will turn off my camera and hand it over to you.
Hi, thanks. So I will just take 2 seconds to share my screen. Let's put it in PowerPoint. Okay, does everyone see my screen? Okay, so yes, my name is Andrea Weise. I work as a biologist on freshwater aquatic invasive species at Fisheries and Oceans Canada in Quebec. And today I will be talking about zebra mussels. And this work was done with the help of many people in our department and outside collaborators. So why talk about zebra mussels? Well, they're bad. They're listed as one of the world's 100 worst invasive species. And for people who are less familiar with them, they're small, they're triangular in shape and have a zigzag pattern. They have high reproduction rates. One female can produce up to 1 million eggs per year, and they can produce massive colonies in the thousands of individuals per square meter. They also have byssal threads, which allow them to attach to many surfaces, for example, plants, rocks, but also boats and docks and infrastructure. And they can also survive out of the water for long periods of time, up to 30 days. As you might know, they also have a lot of environmental, social, and economic impacts. I've listed a few here. They can pose a threat to biodiversity. They compete with native species for resources, and they affect fish spawning areas. They filter a lot of water, and this affects water clarity, and can also affect the growth of unwanted vegetation, for example. They can also facilitate toxic blooms, and when they die, they can litter the shorelines, and their sharp shells can hurt cut our feet, for example. And they can block water intake pipes, and this costs a lot of money to remove and manage. So let's just have a quick recap or a quick look at the distribution over in North America. So this is a map from the USGS, the current situation. And so they arrived in the Great Lakes in 1986, and they really rapidly spread to the rest of the Great Lakes and the St. Lawrence River. Unfortunately, in 2013, they were detected in Manitoba for the first time in Lake Winnipeg, and then continued to spread upwards. And then, although they were present in Quebec and the St. Lawrence River, they were first detected in 2017 in Lake Memphremagog and then Lake Massawippi thereafter. And then recently, they arrived in eastern Canada. This is the furthest east that they've been detected in Lake Témiscouata. And then unfortunately, last year, we also detected them in the St. John River. So Témiscouata is a very large lake south of the St. Lawrence River, and it could act as a source for further spread of zebra mussel in eastern Canada. So what follows is the first look at some of our preliminary data that we collected with our collaborators for primary publication. And basically, the questions we want to answer are, where are they? Where are they headed? And what we can do. So To look at the first objective, which was to better understand the current situation in the lake, in 2022, the province did a baseline inventory of zebra mussels present in the lake. So we did the same again last summer in 2023 with diver surveys at 12 sites at 5-meter depth, where we collected 8 quadrats per site. So here's some of the preliminary data. On your left, you can see a map of the abundances. So the number of individuals per square meter, the size of the circle represents the abundances. And on the right, you have the size classes of the zebra mussels that we collected. So from the map on the left, you can see that they're really present throughout the lake. And this lake is about 40 kilometers long. They're well established. And we came across abundances ranging from like about 15 individuals to 3,500 individuals per square meter. And this is for the 5 millimeter and more size classes. And you can also notice that several sites had really high abundances here. The smaller ones are in purple, whereas the bigger ones are in red. Here, the size classes, again, we can see there were a lot of these smaller mussels, what we call the young of the year, but we did also see these mussels ranging from 1 to 2 centimeters in length, and the longest mussels we saw were 3.5 centimeters in length. So we need to circle back to our provincial colleagues to compare the data between last year and 2022 to see how they've been evolving, but the abundances do appear to have increased quite a bit. We also looked at zebra mussel reproduction. So we sampled for veligers, which are the mussel larvae. So between June and October, our partners sampled at 6 different sites, 2 within the lake and 4 along the Madawaska River, and 3 replicate net tows were done per site. So here's a graph of some of the preliminary results again. These are the abundances per cubic meter. So you can see the peak of in reproduction was really here in August, and the highest sort of values were around 1,500 individuals per cubic meter. And of course, they were highest within the lake, the gray and black bars here. And then of course, and also at the station just downstream below the dam in the Madawaska River. We also detected them at the other sites all the way up to the Edmundston Dam, and later on in the autumn, they were detected on the dam as well. So the question then is, what about elsewhere around Lake Témiscouata? So last summer, early detection was put into place with partners to sample nearby lakes. This was done with several detection methods, which included visual surveys, plankton tows, collector plates, and water sampling for eDNA. So this map shows where we had detections. So the red circles are the visual confirmation. So of course, in Témiscouata Lake, Madawaska River, which flows into the St. John River, and then here in the St. John River at the Edmundston Dam. The orange circle represents the water samples that tested positive for zebra mussel eDNA. So they were detected in the water, in some of the water samples collected in the St. John River. And then here up in Quebec, in January, the province announced the results from the 20 lakes that they had surveyed, and 8 lakes of these had tested positive up here. But we do need to go back to these this summer and validate if the zebra mussels are indeed there. So the question is, okay, where are they headed? How can they spread? So we need to answer that. We need to look at the vectors. So they can either be by natural dispersal or human-mediated vectors. So recreational boating, as you know, can be a potential vector for AIS spread. And this is why outreach activities are so important. And so last summer, several of these activities took place across Quebec to promote the awareness on aquatic invasive species and to wash some of the boats that are present there. So during the cleaning operations, we asked owners to respond to our surveys on their boating habits. So we, with our partners, we collected survey data that included information on the watercraft type, so from simple to complex boats, which will help us assess the risk. You know, simple boats being like kayaks, and more complex boats being boats that have compartments or ballast, things like that. We also questioned them about travel destinations. So from where were they coming from and where were they going to within the next month or so. And we also asked questions regarding boat hygiene practices, so clean, drain, dry. A total of 11 lakes were visited, and almost close to 400 surveys were collected. So I just want to quickly show you the results for Témiscouata Lake and the results concerning the travel destinations within the 30-day period. So here, this map shows the results from the 250 surveys, or close to 250 surveys that were collected in Lake Témiscouata. And you can see the links to 32 lakes and 7 river sites here in gray and pink. And Témiscouata is here in the center, of course. So the lines represent the number of times these lakes were mentioned in the surveys. So I want to draw your attention to the top furthest distances that the boats came from or went to. And Lake Ontario was mentioned in 4 surveys, which is about 760 kilometers away. The Bay James was also mentioned, which is about 650 kilometers away in 1 survey. And then we had 3 lakes that were about 500 or so kilometers away that were mentioned in 1 or 2 surveys. Another interesting thing to look at is maybe which lakes are the most popular, where do the boaters go to more often? And these are identified by the blue circles here. And the top 5 were Lake Pohénégamook, which is just west of Témiscouata, with 12 mentions, so 12 surveys mentioning that they were going there. The St. Lawrence River and 8 surveys. And then south here, Lake Baker in New Brunswick, 6 surveys mentioned them. And then east, Lake Métis in 5 surveys. And as I already mentioned, Lake Ontario. So this type of information could be useful to prioritize where we should be putting our energy for outreach and prevention. Just wanted to show you another second map, zooming into the St. Lawrence area. So the links, you couldn't see these on the other map. Again, in yellow, the Témiscouata Lake, and the blue were the other surveyed lakes. And you can see the different links between these lakes, the direct links and sort of the indirect links. And you can see that they're all fairly interconnected. On top of this, we put the lakes where the province collected water samples that had tested positive for zebra mussel eDNA. So the next question then would be, do we have suitable habitats for zebra mussels to establish? So one quick way to do this is to assess this by looking at calcium in the water. Calcium is important for shell growth and has been used as an indicator of suitable habitats. I put an asterisk here because we know it's more complicated than that. It's not just calcium, but there's also temperature, pH, and salinity that are important. But calcium is a good starting point, and these are usually the values that are used to sort of assess which habitats might be suitable for zebra mussels to establish. So under 8 milligrams per liter, this is considered, very minimal, not enough really, or just limited adult survival. From 8 to 12 milligrams per liter, larvae can develop. Greater than 12, so between 12 and 20 milligrams per liter, we have survival and reproduction. So this is moderate suitability. And then as we get to sort of 20, 25 and more higher values, then these are really very highly suitable areas. So this is some of the data we put together for a zebra mussel risk assessment, where we collected all of the available data on calcium and pH from North America. And just looking at this map for Eastern Canada, you can see the red portions here, the Great Lakes. These are very highly suitable areas with high calcium values, and this is where the zebra mussels invasion started. If we look to southern Quebec, we have some pockets of very highly suitable areas. And here, near the Témiscouata is located here close to Maine and New Brunswick, Quebec. And you can see that all of the Gaspé Peninsula has highly suitable areas. Going into the New Brunswick, the St. John River watershed, also quite suitable habitats, as well as the eastern coast of New Brunswick and Prince Edward Island. So this could be used as a tool to identify areas of concern.
Got about a minute and a half left, Andrea.
Okay, so what next? So I'll just recap quickly. We know that they're established, they can spread, there's risk with the recreational boating, and we have suitable habitats. So the problem is zebra mussels are difficult or impossible to eradicate once established. So what's left? We have to work on prevention. So before I just finish up with prevention, I wanted to play a game with you and see if you could see the zebra mussel. So I'm going to show you some photos of a study that we're doing on the association of zebra mussels with plants. So on these photos, can you see the zebra mussels? So I imagine you can see these two, and I just wanted to point out that they're small, just 1 centimeter. So this is like your pinky finger. But did you happen to see them here? They're on the stems of the plants, and they were hidden here. So this makes you think they can be hiding anywhere on your boat. As you know, they can attach here to the boat, to the propeller, to the trailer. They can also be hiding on your equipment, on ropes and things, but they can also be in your standing water inside of the boat. So we may be inadvertently bringing them along to another lake when we move one boat to another site. So how do we prevent further AIS spread? Well, it's to continue promoting the best practices to prevent their spread. So many Western provinces and US states have put in place a mandatory pull the plug and watercraft inspection and decontamination programs in place. So pull the plug is that you remove your drain plug. And these are really great examples of programs that can help prevent the further spread of AIS. Of course, the Clean Drain Dry, these outreach and education campaigns, they're also really important best practices, and they're really easily accessible to the general public and should continue to be implemented. So I just wanted to quickly review Clean Drain Dry for those that are less familiar. What we mean by clean is that we want to clean the interior and exterior of your watercraft, the trailer and equipment, remove plants, animals, and muds. And we know that if possible, use hot water because this kills invasive species, while low and high pressure water helps remove the invasive species. You want to drain water from the internal compartments, such as the bilge, live wells, ballast tanks, and from your equipment like coolers and buckets. And you want to remove the drain plug. And finally, you want to dry the watercraft and equipment before moving to another water body. So collaboration is really key here between the different stakeholders to prevent the spread of AIS. So just want to finish quickly by thanking all the people that were involved. So the various colleagues from DFO in our department and from New Brunswick, and our outside collaborators, including Annick Drouin and her team from Quebec province, our watershed group, Anne-Alaure Duchesne and her team from the Organisme de Bassin Versant du Fleuve Saint-Jean, and then Charles O'Neill-Sprites from the Société d'Aménagement de la Rivière Madawaska with her team, and Kristen Elton and her team from the New Brunswick Invasive Species Center. So I think we'll continue working together for these common goals, as Sarah Rang had mentioned in her welcoming remarks, in the sense that we strive to enhance our knowledge and want to mobilize action on AIS. So with that, thanks for listening. And I left my email address if you want to contact me later on, and welcome any questions if you like.
Okay.